Hello, everyone. This is the Impact Factor. And a couple of weeks ago, 2 weeks ago, I wrote a paper on why I believe the REGAL trial will fail and SLS, the parent company of the drug, will crash. So, SLS is the company Sellas Therapeutics with their CEO and founder, Angelos Stergiou. I think that's the way you pronounce it. The guy is Greek. And they are making a TEA protein based vaccine targeting a VT1 protein for AML. Now, this is all a little complicated, so I'll explain to you today why I believe their trial will fail, and this is their only asset. So eventually, after that happens, the stock will crash a lot. So first things first, what is AML? Well, AML is acute myeloid leukemia. That's a cancer. It's a cancer that basically happens in your bone marrow, where you produce defense cells for your organism that protect you from other diseases. And when it happens, you stop producing those defense cells. Eventually, you die. You get a shortage of red blood cells, white blood cells, and instead of them, you start producing things called blasts. Those accumulate in the bone marrow and spill into the bloodstream, and eventually you die. This is a very tough cancer to treat because usually what happens is it goes into a relapse, and well, it just comes back. So not a nice disease to have, one of the tougher cancers to treat, and this is particularly interesting why they chose this one. The terms that you also need to remember is CR1 and CR2. These are complete remissions. So first you get AML, they blast you with radiation, radiotherapy, chemotherapy, sorry, well, bad things that make your hair fall out and make you look like Yoda. You hopefully achieve CR1, so the cancer has stopped, has reduced, and you kind of get your healthy blood cells level up. But eventually it returns, and it's usually a matter of a couple of years. So you get your second chemo, so this is chemo 2, and you hopefully get to CR2. And there are statistics on how many people achieve them, and I mentioned them later in the paper. They're important for valuation that we'll be doing. The first problem that I have with this treatment with GPS, that's the name of the drug. The name of the drug is Galinpepimut S, GPS in short. And what this is basically a vaccine for cancer. This is a peptide vaccine, which means that it will be focusing peptide. So what this is supposed to be doing is you have these cells in your body. Would be wonderful if I could change the color. You have these cells in your body. These are the good guys, and they go in and they attack toxins. And what you want to do is you want to inject this vaccine, which is a TAA vaccine, a tumor-associated antigen vaccine, into your bloodstream to teach these guys to attack based on something called a VT1 protein. And the VT1 protein is expressed in those cancer cells. So say this is cancer, this is a bad guy, and he has this little point here, which is a VT1 protein. And what you want to do is you want to teach these guys to attack him and hopefully kill him. The problem with this mechanism is that it doesn't work. So an industry analyst tracking synthetic peptide vaccine found that out of 1,000 examined TAA vaccines, 125 reached phase 1 and 30 reached phase 2, and none ever cleared phase 3 to reach the market. Why is that? Well, there's many different theories, but typically the problem is that people think it is that you are making your body attack itself. So this protein, VT1, is not just something that is only found in these cancer cells. It's also found, if I remember properly, in the liver. So this is your liver, somewhat like this, maybe a little more damaged if you drink a lot. And the liver also has these proteins. And there's a ton of different important things in your body that also express the VT1 protein. And your body has spent years, decades, maybe even half a century if you're an AML patient, because it's usually happening to older people, teaching its cells CD8 and CD4s. I probably should tell you what that is too. Teaching them, those are basically the defense cells, teaching them that do not attack just based on VT1 because it's part of you. It's kind of as if your own organism started attacking your red blood cells. That happens rarely, but it's a disease and you don't want to have that because precisely you don't want your organism to turn against itself. There's an example, a counterpoint to this. It's a drug called Provenge with this organic name. Don't remember, I'm not going to pronounce that. But this is actually a cell-based therapy. So what they do is they take the fusion protein outside the body, and it's not a standalone vaccine like GPS. It's an entirely different type of a drug, and it's actually closer to cell-based therapy than to a TAA vaccine. And this has already happened. There's a ton of other drugs just like this that failed. Actually, there's a chapter. Is this V2? No, this is V1. Okay, I gotta download V2 because there's two versions of this file. One is from earlier, the other one is from the day after. I just added a chapter that showed better what I wanted to convey. And the point is, there have been other vaccines just like this that were also supposed to target a protein in your body, like this one, MUC1, and they looked very promising, and they got nice trial results and FDA breakthrough designations. And this has happened before. There were drugs that looked like, oh my God, this is it, everything's going to change now. Well, it didn't. And this is a pattern that's very consistent in biotech. And this is another point that I want to make. I switched to the V2 of my paper here. There was a very similar drug just for this designation conducted by Otsuka. Otsuka was or is a pharma company, and they made something called OCV 501, which was also a VT1 targeting helper peptide vaccine. So they one of these, CD4, is a helper. So it kind of, when there's a bad guy, it kind of hooks up to it, and he says, hey guys, this is a bad guy here. This is a bad guy, and CD4 goes to him, and it kind of starts alerting the other cells that, hey, attack him. And this is what they chose for the basis of this OCV 501 drug, and it failed. But it didn't fail. That's my idea. That's my thesis. It didn't fail because of the CD4s and not CD8s were used. It failed because the entire idea for making this drug is faulty. Exactly the same thing happened. So I think there was a supposedly a difference in overall survival, but there was no p-value disclosed, and the asset kind of died. And if you think about it, if you have a potentially multi-billion breakthrough drug for cancer, and if it was statistically significant, they wouldn't have just left it there for nothing. And Sellas also already ran this experiment. So they had this drug called Neuvax, Neuvax. It was a HER2 targeting peptide vaccine that was using the same platform, short peptide and a GM-CSF adjuvant. This thing basically makes your bone marrow produce more defense cells. And it was aimed at breast cancer. Pivotal phase 3 present trial, 758 patients, which is much more than what Sellas is doing right now with GPS. That's also important. Randomized double-blind placebo control, and was stopped early for futility. This is also important because some committees review those trials, and based on what they see, they look at the data, they unblind the data so they can actually see whether placebo is doing much worse or the same as the drug. If they see that this is futile, they're going to stop the trial. But sometimes, if they see that there's a clear difference between the drug and the placebo group, they will actually stop the trial anyway and just proclaim it a success. And this has happened, and this is actually a reason why you would want your clinical trial to stop as early, because you want to see that there is such a huge difference between the drug and placebo that you just stop the trial and you just progress with the FDA from there. That has happened in the past, and that is what you want to go for, not the other way around. So here, we'll get to that in a due time. So anyway, they tried this, failed. The guy, there's also I also have a problem with the CEO. So Angelos Stergiou runs Sellas for 14 years now, since its founding in 2012. And this guy has no history of getting any drug approved. So not only do you have a scheme, a designation of TAA peptide vaccines that has 0 drugs approved, so class is 0 drugs. You also have a CEO with a history of not getting drugs approved. And this is kind of, well, just if you were along, think about it as if what you would have to believe that this will be a success. You would have to believe that this will be a first in class drug delivered to you by a guy who never delivered anything. Okay, well, maybe. Let's go on and see what's next. He also worked at Anavex. Anavex is this famous company that failed while they were making a drug for Alzheimer's disease. Cool case study. Should have shorted it. Didn't. I was just looking at it. But I don't think he had too much involvement in Anavex. But it kind of just goes to show what kind of other drug companies this guy has been involved with. Now let's get to the biology of VT1. So it's theoretically the perfect target for the vaccine. The problem is that it is already a normal protein in your body. It's not mutated, it's not foreign looking, and it's hard to make your body attack itself. The entire point of the immune system is to attack foreign objects and to not attack your body. And this is the textbook reason why TAA vaccines underperform. And I found a study that also has identified a second independent problem, which is that some self-tumor peptides don't sit tightly enough in the MHC groove on the self to hold the T cell attention long enough to activate it. So, what the hell does that mean? So there's this tumor here, right? And this is actually pretty cool. So every cell in your body, what it does is it kind of, there's things happening here, right? This is a cell. This is a single cell. And there's things happening inside of it. And the cell kind of takes out parts of those little things that happen inside of it, and it pushes them out to its surface, precisely so that other cells, like T cells, can come over here. Suppose this is a T cell, okay? The T cell comes over here and he's checking out the cell. And by looking just at the surface, you don't have to go inside the cell or cut it up and see what's inside. You just check what's on the surface. And this way the T cell can identify what's happening inside the cell. Now, the MHC, if I remember correctly, was the particular point where those things that happen inside were being exhibited to the T cells. So the problem can be, as this paper here identified, there's a clickable link here, you can check it out. Poor immunogenicity of a self-tumor antigen derives from peptide MHC1 inability and its independence independent of tolerance. Um, they believe that there is not enough time for the T cell to stay on top of the MHC to actually identify the target and to lock into it. So this could be another problem that's happening. I have an analogy here that says, think about it like the MHC molecule is a person holding out their hand, and the peptide is the type of grip they use. And the T cell is the receptor, or another person that is, walking by you and shaking your hand. And if you just give them a lumpy, slimy hand, and you just kind of, barely grip their hand, they're just going to slide off you, and they're not going to stop. But if the handshake is firm enough, they might actually stop for a second and, say, hi, nice to meet you. Who are you? What do you do? That's the point. So even if the T cell can recognize the peptide, it is possible that it won't activate the peptide MHC complex just because the connection is not stable enough. There's also a straightforward escape route. AML is genetically messy, so there's a lot of subclones, and the population is not uniform. So some AML cells, some of these cancer cells, here's some of them, don't be fooled by their smiles, they're the bad guys. You can see it by their eyebrows. AML cells. Some of them have VT1, but some of them don't. So even if this works, and I've already given you 2 reasons why I believe it won't work, I mean, I actually gave you 4 reasons, but biologically 2 reasons. Even if you kill these guys, there's still some cells that don't express a VT1 protein. So these guys will keep on multiplying. And eventually, you're just going to have AML that has no VT1 production, expression. And, even though these guys with VT1 died, these guys are fine and dandy. So you're still going to die. Okay, so let's move on to the phase 2 data, which is part of the Bull thesis, and it's presented as this huge success, which I think is a wrong interpretation. So first of all, this was a single-arm, non-randomized trial. So this means that you had no placebo, so you don't really know how much of the success of the drug is the drug. You don't know how much of the success of the improvement in overall survival was the drug, and how much was it that, for example, you just chose the patients that were better off, that were in better shape than historically. So what you compare yourself here to is you have this group of patients and you give them all drugs, and then you look at historical patients and you see that, wow, they lived longer. Yeah, no shit. The guys you picked historically had, I don't know, were 80 and had liver damage and tons of other sicknesses. And this time you chose a bunch of 60-year-olds that had just gotten their remission. So there's a chance that they fucked up in that way. Then secondly, and this is pretty important, phase 2 was actually CR1. So we talked about what this means. This means that it was the first remission that the patients got. And now the phase 3 went for CR2 patients. I believe that this is because, if I remember correctly, they said that CR2 patients are going to have a higher VT1 expression, which is going to be supposedly even better exposed, which is going to make the drug look even better, work even better than the CR1 patients. Well, the problem is that those patients are way sicker. Usually the patients that achieve CR2, I believe, have a survival of 2 years or something like that. So, yeah. But this also means that, the patients in CR1 compared to CR2 were meaningfully healthier. And this is why this result seems so stunning. And then again, this trial for this phase 2 was super tiny. So you had 22 patients. N of 22. N is like cases or situations or events in statistics. And then you had the 47% 3-year survival figure, but it was based on just 19 patients. And at this size, this is, not that meaningful. So you did have this fantastic median OS not reached, um, and the overall survival was so fantastic, but it was possibly because they chose healthier patients, and possibly, and then again, possibly because it was CR1 and it's not CR2, and because the trial was smaller. So what happens when you have a smaller sample size is you get higher standard deviations. Can I write something? Oh, wonderful. So as your n goes down, your standard deviation can go up. And this is why it's harder to produce. This is why usually as your trials proceed, you move from the first to the second to the third, the number of patients go up. This is pretty cool. So this is as clear of a comparison of GPS to other cancer vaccines that have already been tried and have already failed. So here we have OCV501 and Noivax. Noivax was this drug that was tried by the same company earlier, and OCV was by Otsuka. So target antigen was the same in OCV and GPS. Noivax had a different antigen. The developers were different. The mechanism, the T-cell mechanism, so Otsuka used just the CD4s, just the helpers. Noivax used CD8s, which are the killer cells. They actually do the job. And GPS is trying to use both of them. So, hopefully that works better. Peptide alone, GM-CSV, and here we got other helpers for the production of, for the stimulation of bone marrow. So hopefully the idea is this will work and this will stimulate your bone marrow to produce more T cells. And the point is that the similarity is pretty clear here, that, if CD4 failed and CD8 failed, what's the chance that CD4s and CD8s are going to work together? I mean, it's not really the same as trying to move a cabin, not a cabin, trying to move a closet with your friend. And if you do it alone, you're not going to move it, but if you do it together, you will. That's really not the case here. I don't think there's a clear path that says that this will work because you use those cells together. This is also pretty important. So the immune response. This is some data that was really boasted by Sellas. They said that GPS was hitting a very high immune response, and if you compare them to the other drugs, an immune response is basically that your drug entered your bloodstream, right? This is your bloodstream. It's black because you've been drinking a lot of Coca-Cola, and you give them the vaccine, and your T cells are like, yo, now we attack VT1. This is your immune response. And they were boasting that GPS had a high one. Well, so did OCV and so did Noivax. 100 for OCV and 74 for Noivax. And this is pretty good. And still the drugs failed. So just because you get an immune response, I don't think you're going to get a working vaccine and a provable, meaningful, statistically significant improvement in survival. Then we get to the point with the trial itself. So the trial, REGAL, it's a phase 3 in CR2. REGAL, it has some real problems too. So this trial is for 127 patients, which is, if you notice, smaller than for Noivax and for other vaccines of this type, for other drugs of this type at this stage. Many companies have phase 2 trials that have more patients than 127. And this This is a P3. This is supposed to stop, the trial is supposed to stop when we reach 80 events, which are patient deaths. We are currently, as of 7th, 19th, 26, at 78 deaths. And it's actually taken way longer than we expected. So I think at first, the trial was believed to come to a conclusion at the beginning of 2025, if I remember correctly, and this didn't happen. And then it was supposed to come to a conclusion at the end of 25, and this didn't happen. And now we're kind of waiting, and we're sitting at 76, 78 deaths, and even the CEO is going kind of crazy and talking bullshit about making LinkedIn posts about how this is completely important and the drug definitely works. And I encourage you to go read his LinkedIn posts. The guy is a little funny. The trial is open label, not blinded, so that introduces biases. And the historical benchmark is about 6 months expected survival for the population. OS of 6 months expected. And I believe that why the trial is taking so long, well, there is a couple reasons for that. One is that the enrollment curve was not very steep. So, where's this? There it is. So, this is your number of patients, and this is time. When you start enrolling patients, and say it looks like this, that's a pretty steep enrollment curve, right? So here you got one first patient, here you got the second, third, and now you get 5 and 8, and then you jump to 12. What I think could have happened here was that they were very slow at taking in the patients. So that's one of the reasons why the trial is taking so long. So say this was, I think this trial started in, let's see. Okay, I have no idea where the trial started. I do know that we had 66 patients dead in 2024 and 123 enrolled. So they still haven't had all their patients enrolled at the time, as of March 2024. I do see that enrollment was complete at the end of March 2024. So they got their 4 patients, and the trial began as of August 2020. So this trial has actually been going on for almost 6 years. So here it started in 2020, and only in 24 they reached their goal of 127 patients in 2024. So now we are here, 2026, and so this probably looks kind of like this. The patients are dying, and the people think that, wow, the trial is taking longer than expected. This must mean the drug is working, right? They were expected to only survive for 6 months, and they actually survive much longer. Well, no. Another reason, apart from the enrollment curve, and we don't know what it looks like because they don't tell you. Maybe they're going to tell you after the trial is released. But another reason is that somebody fucked up the calculation of overall survival. So maybe you just got stronger patients. Maybe you got younger patients. Maybe you got patients with less diseases before this. And also there's just statistical randomness. Sometimes, you do a lot of coin flips. Eventually you're going to get a streak of getting tails 10 times in a row. And you do enough of these small sample tests, you will eventually get a group of patients that is going to live way longer than expected. And this is one of my key points, because this is the reason why the stock has gone haywire. If you look at it, so the stock was trading at $2 still at the end of 2025, and now it's at 13. And yeah, people, I think, believe that I was actually going to short it at 4. Good thing I didn't. I shorted it now. People actually believe that because the trial is taking longer, this is bullish. It, in fact, is not. So this is something that is a common misconception. Just because they're living longer doesn't mean A, that the drug is working, and B, that the drug is working. um, so there have been a lot of cases of companies where the phase two was taking longer than expected. This has happened in the past, and nothing came out of it. So hold on, let me find this piece somewhere here. So here are some other examples. So Telik was making a drug that also took longer than expected. This is the name of the drug. I'm not going to read it. For ovarian cancer, and it was taking years, while overall survival was supposed to be 3.5 months. The stock ran up. Smart people shorted it, and it crushed. Genvec was making TF Arrayed, same thing. Novellos NOV002 missed phase 3 endpoints in lung cancer. Exactly the same thing. Cell therapies. And you want to compare this to what happens when a drug is actually working and the effect is actually big. So Roche had a drug in melanoma that was stopped early because it was working so well. You could see the difference between placebo and non-placebo patients. And in this case, because the trial is blinded, it's blinded. Is it not blinded? I think it's unblinded. Um, anyway, because there's a committee that oversaw the trial and decided that it should keep going. I think at 60 events. Um, that doesn't mean that the drug is working. If they knew that the drug is working, they would stop the trial. They just said that, we don't know yet. Let's keep going. That's not bullish. That's expected. Unless you fucked up real bad and you make a drug so bad that it literally kills patients. If you literally make a drug that is so bad that placebo patients, people who think they're getting the drug, are doing better than people who get the drug, that's really not great for you. But if the commission said that you should keep going, that just means that they're not sure. It's not bullish. Onyx had a drug for cancer that was stopped early because it worked so well. Algeta, same situation. Trials that quietly run long past their planned timeline are empirically much more often explained by a misguided starting assumption and not by a hidden winner. Here's another point to the immune response. So, we actually got the data from phase 2 only for 14 patients of the 22 enrolled that had any immune response collected. 8 were never tested, and some of them likely relapsed and dropped out early. So of the 14 tested, 9 were positive in the last in at least one assay, which is commonly cited as 64% success. You can see it very often in their press releases where they're boasting it. But 9 out of 22 is 41 and not 64. And there's no reason to test just 9 to test just 14 patients where you have a total population of 22. That's kind of like saying, you play 10 games of CSGO, you take off your socks during half of them, and you win 3 games here and 3 games there. You didn't. I fucked up the analogy. You just forget it. The point is, you cannot discourage discount some of your patients based on uh, based on whether you could have tested them, because I believe some of these patients could not have been tested. So we don't really know what the immune response is, but it's certainly not 64. It's more closer to 41, and even if it was 64, that's still less than OCV and Neovax, which both failed, even though they had a high response. And this is one of my favorite parts here, valuation. Because whatever you think about the clinical trial or the success of a drug, you should weigh it in against the price. If you're paying too much for this, then, you're going to lose anyway. even if the drug works. And this is one of my main points here for the short thesis. So this was written as of July 5th. We're going to stick to those numbers. I think right now the stock is around $13, which is close enough. So $15 a share here with roughly 186 million shares outstanding. So you get a market cap of 2.8 billion for a company that has one drug. Well, they also have this SLS009, but it's still in phase 2, and they have some stuff in preclinical and phase 1. This is very far away, and I find it hard to believe that there will be a huge premium over this asset when this trial fails, if it fails, right? 107 million of cash, no debt, so an EV of about 2.7 billion. And what we got to think about is, okay, so if the drug works, is it actually going to be worth 2.7 billion? Because 2.7 billion is a lot. We don't think about it nowadays because we get companies racing for $2 trillion valuations in a day, and we kind of don't think that 2.7 billion is a lot, but it is. And the market doesn't like it when there is a overvalued asset for a long time, which we don't see right now because we kind of have a bubble. So you have an addressable population of roughly 20,000 new AML cases annually in the US. So around 12,000 of those will reach a CR1. Then you're going to have a relapse, and only a minority of the relapse will get CR2. So some of them are also going to be transplant eligible. So what you need in AML is a transplant of bone marrow, and you hopefully are going to start reproducing that new bone marrow. It's going to be accepted by your body, and it will start producing T cells. So with this, you get about 1,000 to 1,300 patients in the US, or maybe about 1,500 and 2,000 at a steady state. So that's basically at any time, right? Because these patients are still going to die. This drug is not going to be a cure. It will be a treatment. So, you can kind of give it to somebody to extend their life, but this is a very tricky cancer and it's still going to get you. So you're going to have about 1,500 and 2,000 patients at a steady state. And if you count in the other areas, which you got to exclude Greater China because they have their license, they have their license, they're licensed away or something like that. So you're going to get about 3,000 to 4,500 steady state. Then you think about how much do you want to price the drug at. And there is already other therapies similar to this drug. So something that you can realistically get here would be around $150,000 to $200,000 a year. I put in my model actually the higher numbers of this reach. So this is a very bullish valuation right here. So I put in 4,500 patients at all times and a drug price of $300,000 per patient per year. So this is really big. So suppose this trial ends this year and they immediately submit a BLA and it's approved and they realistically start selling in 2027. I assumed a very, very rapid adoption. As you can see, I'm assuming 100% market penetration by 2029. This is a great result, because I want to stress test what I'm saying. This is what you get: $500 million in the first year, $1 billion and $100 million next year, and by 2029 you basically get $1.5 billion a year, and then it kind of grows. I assume the drug growing at 3% for inflation purposes, so this is why it peaks around, I think, $2.5 billion by 2047. I assume the drug patent is going to be last for 20 years. So profit margins of 39%, pretty nice, and we get a double from here. So $29 a share, best case scenario. So just a double from here. And this is, as good as it gets. That's what I get. And that's my main point. So on top of this drug that has a zero in class, a CEO that did no drugs before that, two other drugs that failed, OCV and Noivax, that were extremely similar to this, an immune response which doesn't really matter here because these drugs had higher immune responses and still failed. What other arguments are there? A great phase 2 data that was in a small population and in CR1. Can you stop doing this? I fucking hate this pen. Oh yeah, okay, there we go. Phase 2 that was good but questionable, and it was in the first recession and not the second one, a remission, not recession. And you have a historical case where drug companies take longer than expected to produce data and it fails. So you take all of that in, I believe that's a 5% probability event that they will get this drug approved, that this works. 5%, and I think this might be on the generous side because I want to give them the benefit of the doubt. The what if this is a cure? Oh my god, bro, they're geniuses. They're a bunch of geniuses. What if? I want to believe it. I really don't. And then I give them a miracle capitalization scenario on their asset, and you get a double from here. So do you really want to risk the 5% probability of getting this drug approved? And then, I don't know, this is maybe a 10% for this type of capitalization with these numbers of patients and this cost per patient. At 3 billion, this is just a really expensive company with no other assets, management that has zero success in the past, and it's just expensive. You know, so I believe that when this drug fails, this will go to around cash value, which is I see around 58 cents here per share, plus possibly a dollar, a cope dollar for hope that this SLS009 is going to work. So I believe that this stock has a long way to go down back to $1.50, where it was just a year ago. Remember that this stock kept going up because people think that, hey, this trial is taking longer than expected, therefore it must work. Well, sorry, not really. And this is a crowded short. What I mean by this is that the stock borrow rate, which is how much you have to pay to stay short this stock, is 150%. That's a lot. That means that smart money is piling in to short this. And do you really think that the guy with no history of approved drugs, no drugs in this class, that he's going to be the one to do it, and you are the one to see that this is really the opportunity to take you out of the projects? I don't think so. You know, there's a lot of smart people there doing exactly what you and I do, looking at the stock market 24 hours for a week, and they fail. And this is why the borrow rate is so hard, because to many smart people, this is an obvious short. Knowing where the fools is, who the fool is. So this is one of my favorite parts. The stock has a Reddit community, and these people are literally brainstorming what they're gonna name their children after this drug makes them billionaires. Yeah. Somebody claimed that he's naming his son GPS. Okay, like I like to know who I'm betting against. You know, if I'm betting against Ken Griffin and Paul Tudor Jones, and on my side I have my buddies from college that are laughing about how they're gonna be billionaires. Yeah, I don't know, man. Maybe that's a little too much. So this is a summary where I land, and this is pretty much it. You can find this thesis on my GitHub. This is my GitHub right here. I have a repo here for biopharma. Here you have the model and you have the papers and some other stuff too. So feel free to follow up and to check it out and to read it yourself. I am very happy to hear your comments, as long as you don't call me a piece of shit like most people on X do. I will maybe text you back, not sure, because I don't really care. And most of the criticisms that I got are bullshit. So it's just people calling me an asshole and bringing up data that doesn't really matter. Yeah, so there's that. This model, these papers are for free, models are for free on my GitHub. If you're long this stock with 100% of your portfolio, please cut it down. Please, just please. Thank you for watching. At the end, I'm going to show you a clip that I really enjoyed from a pharma expert, Martin Shkreli, and what he said about the post of the CEO on LinkedIn. And thank you for watching. Subscribe to the channel, follow me on X, and give this video a like and a comment, and see you in the next one.
What slower event accrual actually signifies? The noise is slower event accrual means the control arm is surviving too long. What the? This guy's retarded, isn't he? Oh, he's Joe Springer. My takeaway from that is this guy is legitimately just doesn't know clinical trials. Well, he's going to learn.